So what we're starting on today is Chapter 3 on cells. And what we're going to be focusing on for today's lecture is kind of an overview of the study of cells as well as the tools. So the tools that we use, we're going to talk a little bit about microscopy. We're going to talk a little bit about different general cell types, so prokaryotic cells versus eukaryotic cells. And then we're going to actually start by talking about the membrane, the cell membrane that's on the outside of all cells. So we're not going to really get into the components of a cell today. On Monday, we'll get into the main components of a eukaryotic cell, and then next Wednesday, we'll start talking about some other structures that are in it. But really, the chapter on cells is all about the components that make up the cell. So cells are units of life. They are the smallest collection of matter that can function independently. So there are single-celled organisms out there, and if you do take a microbiology course, you're going to be taking an entire course about single-celled organisms, bacteria for the most part. So you're going to be talking about prokaryotic cells in microbiology. We spend most of our time in this course talking about eukaryotic cells, a little more complex cells, and we kind of focus more into the multicellular organism. But there are lots and lots, and the majority of organisms are just single cells. In terms of living matter, that's what we consider the smallest thing that can live. Below that, we're talking about components of a cell and not actual living organisms. So all organisms are made up of cells. They all consist of cells, either a single one or multiple ones working together. And within each of those cells, there are coordinated biochemical activities that carry out the different functions of life. So in future chapters, we're going to talk about processes like photosynthesis, like cellular respiration, which all occur inside of cells and require some of the structures that we're going to talk about in this chapter in order to function. Basically, this chapter, we're going to take all the biochemicals that you learned in the last chapter and see what roles they play within a cell to allow a cell to do these different functions. So cells are fundamental units of life. Cells are, in general, fairly small. So how do we study them? Yeah. So some of you have maybe started doing this in your lab class, if you've had lab already. A lot have done lab 4 already. But we need to use tools to help us study cells. There are some cells that you can see without a microscope. Frog eggs, chicken eggs, a lot of egg cells, a lot of eggs are large enough to see. They're large single cells. There are some organisms that have large cells. So there are a number of algae that have large cells that make them up, and you can actually see their individual cells without a microscope. But most plant and animal cells are a little bit too small to see with the unaided eye. So this chart kind of shows you general size things and what we can see with different tools. Everything to the right of this red line you can see with the unaided eye. So you can see ants pretty easily. You can see larger things. Humans would be up here. You can see frog eggs. But plant and animal cells, bacterial cells, cellular components, those are all smaller than you can see with the unaided eye. So you might need something called a light microscope. This orange line represents a light microscope to see those. But then there's even things that are smaller than a light microscope can see, things like viruses, proteins, even small molecules you might want to look at. And those can be seen by a type of microscope called an electron microscope. And we're going to talk about those different kinds of microscopes as we go along. So microscopes allow biologists to magnify structures so that we can study them, because otherwise they're too small. But you'll see in your lab, you can only gain so much information from a microscope image. You can magnify things, but a lot of these structures are actually clear. So you might actually play around with this in your lab. I don't know if you'll have enough time in that lab. But we use things like stains to actually highlight different areas of the cell and allow us to get a better view of things. But I'm going to show you briefly some of the images that you can get with a microscope and what kinds of questions you can answer with the different types of microscopes. So here's what one of the earliest microscopes looked like. looks nothing like the microscopes we use today. It was more like a magnifying glass is today. This was in the 1600s. This scientist named Leeuwenhoek developed this microscope. It has a single lens. It had a pin where they would put the specimen that they were interested in looking at the tip of the pin, and then they hold it up to the light, and they could adjust the little screws to focus it or to move the object around. And it was able to get a little bit of magnification, probably only 5 or 10 times magnification, very small amount of magnification with the single lens. Nowadays we use much more complex microscopes, and I'm going to show you some pictures of those and pictures from those as we go along. Modern microscopes come in several different types, and each one has a different advantage to it, and we're going to talk a little bit about those before we start getting into more of the components of a cell. So there's two real categories for microscopes: light microscopes versus electron microscopes. And we're going to talk briefly about two kinds of light microscopes: compound light microscopes, which is the type that you'll use in your lab course, and confocal light microscopes, which is just a different type of light microscope, but very similar to your compound microscope. And then electron microscopes, there's two different kinds of electron microscopes: a transmission electron microscope and a scanning electron microscope. And both of those will get different types, give you different types of images that will allow us to get more information. And honestly, a lot of the information we have about cells has been determined through this kind of microscopy, electron microscopy. It's one of the best ways to see very, very, very small structures in cells. And we'll see why that is in a slide or two. Okay, so first off, let's talk about the kind of microscope you're going to use in your lab, and what is a great advantage to it, and what kinds of things can you see with it. So a light microscope, what it does is it shines a light, like it sounds, through a specimen. So you'll have kind of a light bulb of some sort. You know, when I first started taking biology, even throughout college, we just used the window even, and a mirror. But they have lights now, most of them. It shines it through a sample. That light then goes through one lens, and then it goes through a second lens before it gets to your eye. And each time it passes through a lens, it magnifies the image. So that's called a compound microscope. And that's what this is, and that's what you'll use in your lab. So it magnifies the image as it goes through the objective lens, magnifies it again as it goes through the optical lens, and by the time it gets to your eye, it'll be much larger. You'll look at things anywhere from 40 times magnification up to probably 1,000 times. You might, in some of the fancier microscopes, get a little bit larger magnification. The largest magnification a light microscope can get to is 1,600 times, so 1,600 times magnification seems to be the limit of getting a nice clear image from a light microscope. Light microscopes will generate a true color view of living and preserved cells. So you will actually be looking at a living cell in your lab. You will put a leaf of an aquatic plant called Elodea on a microscope slide, and you'll take a look at it, and you'll be able to see the green chloroplast. You'll be able to see the color of the cell. You might actually see little brown things growing on it, because we took those, we got the Elodea from the ponds on campus. And so there's lots of other organisms that might be growing in and around it and in the water. You might see little rotifers feeding on the Elodea. So there'll be these little clear blobs with these really crazy moving mouth parts actually feeding on them, or you might see euglena in there as well. You might see other aquatic organisms. And you can see them moving around because they're alive. You haven't killed them to look at them on the slide. And you'll be able to see their true color, which, for a lot of organisms is clear. But for plants, they have the green chloroplast. You'll be able to see the green chloroplast in them. So this is an image you might get from a light microscope. A confocal microscope is a little bit more complex. It still uses light, still shines light through an object. But what a confocal microscope allows you to do, it actually allows you to focus on different focal points within a sample. So sometimes samples are really thick, and you want to get an idea of what the top of the cell, what the middle of the cell, what the bottom of the cell looks like. Confocal microscope will allow you to view the cell at many different levels, so many different focal points, and then it allows you to merge those images together. It'll take an image at different focal points, merge them all together, and you can get basically a 3D view of a slide. It also usually works really well with fluorescent imaging, and you can actually put these fluorescent stains into the cells and highlight different regions. So one thing we often do, we'll stain for things like nucleotides, and that'll highlight the nucleus and highlight any RNA that might be in the cell. We might stain for lipids, for instance, and that'll highlight all the membranes that are within the cell. You can actually put in these different stains with these different fluorescent dyes, and you can look at different biomolecules found within the cell, and that'll give you an idea of some of the components. Most of the components in the cell are clear. If you scrape off your cheek cell, and you could easily look at it under a microscope, it just looks like a clear blob. It isn't until you put some kind of dye on there to highlight the different biomolecules that you'll actually be able to see anything in it. But when a cell does have color, you'll be able to see it with a light microscope. Okay, so light microscopes can magnify up to about 1,600 times. Things like staining can improve the resolving power, but this is about, or the contrast and things like that, but this is about as big as they can get. The other type of microscope is an electron microscope. And rather than light beams, which are pretty big waves, an electron microscope uses beams of electrons. And electrons travel in a much, much narrower beam than light does. So just imagine you're looking at the surface of something and it has this very finely detailed surface. If you're shining a light beam on it, this whole surface will bounce back and you won't be able to see any of the details. Whereas if you're shining an electron beam at it, you're going to get, the electron beams will hit into the fine crevices on that cell, and it'll give you much, much finer detail. So because of this, because you're using electrons rather than light beams, you can magnify an object incredibly much larger. So you can magnify something now 250,000 times, up to 50 million times its actual size. So it's pretty incredible. So the 50 million times is with what's called a transmission electron microscope. The 250,000 was with a scanning electron microscope. So here are two images. Here's something from what's called a transmission electron microscope. That allows you to see cross-sections of organisms. It'll look at internal structures, and you now get these very fine detailed views. You can now magnify this image a fair amount and look at just one organelle from within the cell and get really good detail of that. This is what's called a scanning electron micrograph. So the scanning electron microscope looks at the cell surface. So you have internal structures versus cell surface in the scanning microscope, and that allows you to get fine details of the surface of an organism. However, one thing you're going to see written next to a lot of these images that are done with a transmission or a scanning microscope is false color. The reason for that is that the images produced by these microscopes are interpretations of electron beams that have bounced off the object, and all that the computer does is turns this into a black and white image. So you don't get true color. You don't actually see the color of the specimen. So they colorize these afterwards. They add color back to them. And imagine how difficult it would be to get an image of a specimen that's moving. You can't have a specimen moving if you're using an electron microscope. So you have to fix the object, and the specimen needs to be dead. So you can't look at something that's alive, right? So electron microscope's much, much more magnification, but you can't look at something that's not living. Okay. All right, so just to kind of go over this again, but what is the advantage, do you think, of using a light microscope? So I said electron microscopes have all these great advantages. Why would anyone still use a light microscope? besides the fact that it costs a lot less. Okay, last few responses. Looks like most of you were paying attention enough. That's good. All right. So the answer would be living cells, like most of you said. So light microscopes can still look at living tissue. Electron microscopes cannot. So light microscopes are used in a lot of fields. My field, we used them a lot, mostly because we didn't need to magnify things 50 million times. I only wanted to look at large-scale structures, and so a light microscope was plenty for that. We even used things called dissecting scopes, which only magnified things about 4 times, and that was plenty. Also, we didn't want to kill the things we were working on. So if you want to look at something and not have to fix it, this is the best way of doing it. The electron microscope is going to magnify it more. It's going to have higher resolving power because that's why you can magnify it more. You get more detail within the object. And contrast, which is the difference between light and dark, that's really enhanced by staining techniques. And it's actually ultimately not all that different, but probably a little bit better in an electron microscope because you're able to stain using various metals, which you wouldn't normally use in the light microscope, and allow you to change the reflectivity of that cell to electrons. So we're going to see images from both these types of microscopes. I'm actually going to show you some videos later on when we get to, especially the mitosis chapter. I'll show you a video of a cell dividing that was actually taken through a microscope. You can attach a video camera to a light microscope, and you can record these organisms actually doing different things. So you get some really neat footage. So a lot of work is done with that. Okay, so light microscopes, you can view living cells, and you can view true color. Electron microscopes, you can view things in much, much more detail. So a lot of the stuff that we will show you over the slides probably next week are electron micrograph images. Okay, so before we get into details of cellular structures, we need to talk briefly, just briefly, about categorization of organisms. So you know what we're studying in this class. This is something, if you took 10, 11, you spent a lot of time talking about taxonomy and biodiversity, and you learn about how things are categorized, how we categorize biological life. And you'll learn things like kingdom, phylum, class, order, family, genus, species, all those kind of categories that allow us to name different organisms and place them in categories based on their relatedness. We're not going to really focus on that in this class because we're so focused on the internal structures of an organism. We're not concerned yet about the differences between different species. But we are interested enough in the large-scale differences between what are called domains, which is even larger group than kingdom. There are 3 main domains of life. There are bacteria, archaea, and eukaryota, or eukarya. And the most recent evidence actually tells us that archaea and eukarya are actually more closely related than they are to bacteria. Bacteria kind of sits on its own. And that's what this tree of life shows you, divergence from a common ancestor. So bacteria diverged from archaea and eukarya earlier from the common ancestor. So they diverged first, and then archaea and eukarya split more recently. So that tells us that eukaryotes are more related to archaea than they are to bacteria. However, a lot of cellular structures and a lot of organization of cells is more similar between bacteria and archaea than they are to eukaryotes. So we're actually going to lump these two together today into prokaryotic cell type and talk about what that means versus the eukaryotic cell type, which we'll get into next week. Most of our course is going to be on this domain, the domain eukaryote, which humans are a member of. If you take microbiology, you're going to spend your time talking about this domain, bacteria. You're going to learn a lot of the same things that we learn in this course, but you're going to learn how bacteria do it. So we're going to learn how eukaryotes generate energy from food. We're going to learn how eukaryotes can become autotrophs. They can absorb energy from their environment. We're going to learn how they divide, how they reproduce, things like that. In microbiology, you learn all those same things, but about bacteria. So we're not going to spend much time talking about bacteria. I'm going to have one slide on it that tells us some of their major features, and then we'll move on to eukaryotes. All right? So even though we know these are more closely related, we're actually going to lump these two together, which is not quite correct, but it's easier for us now. And then we're going to focus on those. So what does a prokaryotic cell look like? This is a bacteria cell, and this is your typical prokaryotic cell. So bacteria and archaea are both prokaryotes. The major thing that means is that they have no internal membrane system. So we're going to look a lot at the membrane system of a eukaryote. The only membrane found in a prokaryote is the membrane surrounding the outside of the cell called the cell membrane. So the cell is surrounded by a cell membrane and cell wall. So in an analogy to a prokaryote, it's like a studio apartment where you have no walls. It's a studio that goes even so far as to not have a separate kitchen or bathroom. It's just one big room where everything happens. Whereas a eukaryote is more like a multi-bedroom apartment that's got lots and lots of rooms, lots and lots of divisions within the cell. So a prokaryote is one big room surrounded by a cell membrane and a cell wall here. Inside that room is DNA, there's going to be genetic material, and we call the region the genetic material is found in the nucleoid, but it doesn't have a distinct place. It doesn't have a membrane of any sort. It's like if you had a studio apartment and you put down a rug and you said, this is my bedroom and over there is my living room, you could easily swap places and there's nothing stopping you from doing that. It's the same idea. Wherever the DNA is, that's the nucleoid, and it can move really easily. There are also these little dots in here are ribosomes. We're going to talk about what those are in a eukaryote. They do the same thing in a prokaryote. They're sites of protein synthesis. They're the enzyme that's going to catalyze protein synthesis. Right, so really, all I want you to know about prokaryotes for this course is that prokaryotes have no internal membrane, and that's pretty much enough. So all the structures that we're going to talk about in a eukaryote, the nucleus, the mitochondria, chloroplast, all those things that have membranes around them don't exist in a prokaryote. All right. So here's what a eukaryotic cell looks like, and Monday we're going to get into some of the details of what all these structures are. Eukaryotic cells have lots of internal membrane, so they divide their internal structures into units called organelles. So just like in a multi-bedroom apartment, you have a kitchen, which is responsible for cooking meals. You have a bedroom, which is responsible for sleeping. You have a bathroom, which is responsible for waste disposal. You have a living room, which is responsible for playing or Jenga, whatever you do in there. I don't know. You've got different things that your different rooms are optimized for, right? Same thing happens in a cell here. You've got a nucleus, you've got mitochondria, you've got lysosomes, you've got Golgi apparatus, you've got all these different structures, each of which has a different function, and each of which is optimized for that function. And we'll get into those details on Monday. The DNA, instead of being just randomly found in a region called a nucleoid, actually has a housing. It has something called a nucleus. The nucleus has a membrane around it which protects the DNA and keeps it in one location within the cell. So we'll talk a lot about this and all these different labels that are on here. Eventually you'll know what these all mean. We'll really talk about it on Monday. So just like a prokaryotic cell, a eukaryotic cell is surrounded by membrane as well. It's surrounded by a cellular membrane, but in addition it has a lot of internal membrane. So before we get into any of the details of the stuff that's in the cell, I want to spend a little bit of time talking about the membrane and what a membrane is composed of, and the structures that are found within the membrane and what role they play in protecting the membrane and maintaining its structure. So this is another graphic like the one I showed you in that first question of a biomolecule which we briefly mentioned in the last chapter, but we're going to spend a lot more time talking about in this chapter, called phospholipid. Phospholipids, remember, have a phosphate group on one end, and that phosphate group is charged, so it is polar, and it has fatty acid tails on the other end, which are nonpolar. Other words you'll see throughout this chapter and kind of used interchangeably with the word polar and nonpolar are hydrophilic and hydrophobic. Hydrophilic means that it will bond with water. Hydrophobic means it will not bond with water. I'm not overly fond of these terms because they kind of give you the impression that hydrophobic things are afraid or repelled by water, whereas hydrophobic things love water. In some ways it's correct, but in some ways it's not quite correct. Hydrophobic things are more indifferent to water rather than scared of water. They don't have charges, so they're not going to react with water. Whereas hydrophilic things they do have charges, they are going to be attracted to it. So I really do like the word nonpolar and polar, but you'll see I use both of them as we go through it. So anytime you hear the word hydrophilic, you should think polar, hydrophobic, you should think nonpolar. So cell membranes are composed of phospholipids, and in the membrane, the phospholipid is arranged into a bilayer. So you'll have these phospholipids arranged like this, with the head pointing up and down, and in the center you'll have the tails. So just looking at these different regions, this central region here where the tails are found, it's all composed of your fatty acids. This is going to be non-polar, and you're going to have polar sides on the top and the bottom. So hydrophilic, hydrophobic. The outside is polar, the inside is nonpolar. So how this acts as a membrane, let's imagine you have something like an ion like sodium, and sodium is attempting to make its way through the membrane. Why do you think it will be difficult for sodium to make it all the way through the membrane?
It would have to get through a nonpolar substance.
Okay, it has to get through a nonpolar substance, which in itself is not the real problem. What's going to stop sodium from going through the nonpolar substance? Anybody have any idea what might happen to sodium as it gets to maybe this point right here in the membrane?
It's stuck to the polar.
All right, yeah, it's not that the nonpolar stuff's going to repel it or the nonpolar stuff is not going to allow it to go anywhere. What's actually going to stop it is this. So this part of the phospholipid has a charge to it. It's polar, right? It's got a negative charge and a positive charge. And this sodium is going to be attracted to that negative charge. So it's going to pull it back up. So kind of an analogy to this is if you have you have a balloon with air in it, you try to push it down in your swimming pool, it's going to pop right back up to the surface, right? You can use a lot of force to push it way down, but it's going to pop right back up. So unless sodium can make it more than halfway across, and if it gets down here, then it'll be attracted to this phosphate, it's not going to be able to get very far. It's going to be turned around. So it would take a lot of energy for that sodium to randomly go through the nonpolar region. Things that are nonpolar, so if you had something like oxygen, which is nonpolar, do you think it's going to be able to go through the plasma membrane? Can you think of anything that might stop it from going through? If you can't, it's okay, because there isn't anything. Right? So because this has no charge, it's really indifferent to the charged things, and it doesn't have any problem with the non-charged things. So oxygen can actually go right across a membrane without any problem. But things with charges are going to be turned around. So the phospholipid bilayer is really good at preventing charged molecules, so things that are polar, ions, from crossing this bilayer, right? So if I want to get something that's polar across the membrane, what I might need is a passageway that has polarity to it. So I'll need to build a tunnel where the inside of the tunnel is polar, so now the sodium could go through this just fine. So that might work, right? Have a tunnel to allow it to cross this nonpolar region encased in something that's polar. What those tunnels are built out of are proteins. So membranes are composed of both phospholipid bilayers and proteins. So these purple blobs here represent proteins that are embedded in the phospholipid bilayer. And one of their roles, and we're going to look at some of their other roles later, one of their roles is to provide passageways to allow polar things and ionic things to pass through the membrane. Right? So this is called the fluid mosaic model. A model in science is a hypothesis. It's a proposed explanation. It's something that is testable. There actually have been a number of tests on the fluid mosaic model that have supported it. It's something that is very hard for us to just directly see. We can't just take a picture and have it look like this. This is a cartoon. It's not a picture, right? It's very hard to see the membrane. But there have been a lot of studies that have supported this. The model that was popular before the fluid mosaic was called the sandwich model, and it had the protein sitting on the top and sitting on the bottom. But biochemists actually showed that the proteins that are associated with the membrane, a lot of them have large hydrophobic regions, which means they have large regions that are made of nonpolar amino acids, and something that was designed that way would not be able to just sit on the surface of the membrane. It would end up sitting across the membrane. So we now know that this is the way it looks. So it's fluid mosaic. So the mosaic part is that proteins are embedded in the membrane. The fluid part is that it moves. So membranes are fluid. Phospholipids in the plasma membrane can move within the bilayer. They can move left and right fairly easily. So two neighboring phospholipids can swap places. And the proteins within the membrane can also move around. So most of the lipids and some of the proteins will drift laterally within the membrane. However, it's very rare for phospholipids to flip sides, for the outside phospholipid to go inside and the inside to go outside. Why do you think that is? Why won't this phospholipid switch places with this one very easily? What's preventing that? Yeah, so just like the ion going through the nonpolar region, this polar phosphate would have to go through the nonpolar region, and it again would be pushed back up. So side-to-side movements happens 10 to the 7th times per second, so 10 million times a second, whereas flip-flopping is about once per month. So it's a very rare event. Very rarely happens. It happens occasionally, but it very rarely happens, right? And that's due to the nonpolar region in the center here. So the second kind of part of this graphic here talks about how your cells can change the fluidity of their membrane. So having a fluid membrane is important for cellular function. There's a lot of stuff that requires proteins to be able to move around within the membrane. Do you have a question?
Yeah, why is it difficult for something polar to cross something that's nonpolar?
Because it will be attracted back to the polar side. So this.
So it's more about what?
Yeah, it's
next to it.
Yeah, it's because it's next to other polar things. It's going to be hard for it to get away from those polar things. It's always going to be pulled back towards them. Yeah, so it's the combination of polar and nonpolar. Yeah, on its own, nonpolar and polar things just won't interact. They'll be fine. It's that the only polarity is up here, and it has this great space where it's going to be always drawn back to where it came from, right? Okay, so the second set of pictures here talks about a fluid versus viscous, so a liquid versus more of a solid surface. Your cells can actually change their fluidity based on the hydrocarbon tails on that phospholipids. Remember, just like in fats, you can have unsaturated hydrocarbon tails versus saturated hydrocarbon tails. So maintaining a fluid membrane is important for cellular function. So let's imagine you were a fish in the Arctic. Which type of membrane do you think would be more advantageous to a fish living in very, very cold climates? To have unsaturated fatty acid tails or to have saturated fatty acid tails?
They want solid so it doesn't freeze, right?
All right, so what do you think will happen to the ability of the cell to function normally if their cell surfaces freeze solid? So remember I said that it's important for cells to have fluidity so they can move things around in their cell surface. So if it freezes solid, that would actually stop cell function. So which form of this would actually maintain fluidity in an Arctic fish? Yeah, this one. So this actually would make it so that even in cold temperatures, their membranes are still a little bit fluid, right? So how about a fish in the tropics that it lives in really warm temperatures? Which of these do you think theirs might look more like? Yeah, it might be more like this so that it doesn't get too fluid and start falling apart. So they want them to be fluid, but they want a certain level of fluidity. So this form would make it more fluid. So something in a cold climate might have this to maintain fluidity, and this makes it more solid. So in a warm climate, you might have that so that it maintains kind of some degree of solidity to their membrane. Right? Something else that maintains fluidity and also maintains the viscosity of your membrane is cholesterol. So cholesterol, we usually hear a lot about the bad parts about cholesterol, but cholesterol does play an important role in our cells. It is found embedded in animal cell membranes, and what it does is it creates these things called lipid rafts. It holds phospholipids together, which actually maintains some kind of structure on that membrane, keeps it slightly viscous, but it also, because it's doing this, it's pushing some of the phospholipids apart, so it maintains fluidity as well. So cholesterol helps to maintain a certain amount of fluidity in your membrane, keeps it from getting too fluid, keeps it from getting too solid. It kind of maintains optimal fluidity. So without any cholesterol at all in your cells, you'd have a lot of problems. So you need some of it in your cells. Our problem is that we tend to get too much through our diets, and it starts causing problems elsewhere in our bodies besides just in our cell surfaces. Right. Okay. So there's just a few more things that I want to go into today, and then we'll leave a lot of this for next week. So there are a bunch of different proteins that are associated with the cell membrane, and we're going to talk a little bit more about these in the next few lectures. So I just want to kind of briefly outline the types of proteins and some of the terms that you'll hear throughout this text. There are things called peripheral proteins, which are proteins that are bound to the surface of the membrane. So you might have a protein that's up here. And a peripheral protein, do you think it'll be polar or nonpolar if it sits on the cell surface?
Polar.
Yeah, so the cell surface, remember this part up here is polar, so any protein that sits right on the cell surface is probably going to be polar as well. There are integral proteins like the one I drew here, which will actually penetrate into the nonpolar hydrophobic core of the membrane. And integral proteins that go all the way across the membrane like this are called transmembrane proteins. So some proteins might only go partway into the center, and some will go all the way from one side to the other. Any kind of integral protein is going to have some kind of nonpolar amino acid to make up the part of it that is in the inside of the membrane. So the hydrophobic regions of integral proteins consist of nonpolar amino acids. So in this protein, that's a transmembrane protein, the amino acids making up these regions are going to be polar, but the amino acids making the outside of this region is going to be nonpolar. The inside of that channel protein is going to be made of polar amino acids. So having the right combination of polar and nonpolar amino acids is going to provide this protein with the function of allowing ions and things to go through it. We're going to talk about these channels and how they can actually allow you to regulate what types of molecules go in and out of your cell by opening and closing different channels. They're all created by putting together the right combination of amino acids. Right? Okay. All right, so what we're going to start on Monday, before we get into some of the details of the different components of a cell, is I'm going to review this again, just because this will be very important as we keep going through the semester. There are many different types of membrane proteins. So there's the one that we talked about, which is a transport protein, allows passage of ions and other substances, of polar substances through the cell. There are also proteins embedded in the membrane that will act as enzymes, and we'll talk about those a few times as we go through the few chapters, especially when we get to photosynthesis and respiration. There are proteins that allow cells to recognize things, recognize themselves, recognize foreign objects, to recognize food sources. There are adhesion proteins, so proteins that allow the cell to stick to things. That also allows for movement, usually, of cells. And there are receptor proteins that allow your cell to respond to stimuli. So especially when we get into that group of slides that I called 3A, where we talk about cell communication, these receptor proteins are going to be very important in taking signals from the outside of the cell and transporting them to the inside of the cell. Okay, and this is kind of what they look like, those different kinds of cell membrane proteins. So again, we'll start off and review this again on Monday, and then we'll go forward from here and start looking at specific things that are found on the cell surface, as well as all the components that are found within the cell. And that'll kind of round out chapter 3. All right, so that's it for today.